Today we are going to share with you our preliminary analysis of the effects of environmental factors on zoonotic emergence, where we looked at environmental drivers on infectious disease. My name is Hannah Elvington.
My name is Shreya Simha.
And my name is Meredith Welch.
When observing biodiversity, there are two hypotheses that correspond, both the dilution and the amplification hypothesis. The dilution hypothesis is that richer biodiversity reduces human exposure to infectious disease. An example of this might be Lyme disease, which means if there are more hosts, then there is less spillover to humans. The amplification hypothesis is that increased biodiversity will increase the risk of human exposure to infectious disease. There has actually been no empirical evidence to be used as an example for this, but through our research project and articles that we have read, we have noticed that biodiversity is context dependent. We looked at different diseases because we wanted to see if the mode of transmission had any effect on biodiversity and land cover change. Originally, we started to look at the hantavirus. However, we also wanted to look into other modes of transmission. Therefore, we also studied dengue, leptospirosis, and leishmaniasis. The hantavirus is a vector-borne disease which is spread through air transmission or ingested droplets from rodents. Dengue is also vector-borne, which is spread by mosquitoes through their bites. Lepto is a waterborne disease, which is spread when a mammal urinates in the water and contaminates the water source, which is then ingested by humans. Leishmaniasis is vector-borne, which is spread through the bite of a female sandfly. Additionally, we decided to study Brazil between the years of 2007 and 2017. We chose 2017 as our end date because there were the last prevalence rates of the hantavirus. Overall, our research objective was to evaluate the association between land cover changes and the occurrence of these zoonotic diseases, and also to describe environmental contexts that may support both the amplification and the dilution hypothesis for disease emergence.
So for our study area, we looked into Brazil because we were able to find the most data for Brazil. We conducted a state-level analysis which consisted of counting for 27 of Brazil's states from the years of 2007 to 2017. Brazil had an adequate notifiable disease surveillance system for all the areas with rapid environmental change. We were able to find case count and population count from Brazil's Ministry of Health for hantavirus, dengue, leptospirosis, and leishmaniasis. With this, we were able to estimate a prevalence rate per 100,000 people for these diseases. Since we were looking at land cover change, we were able to obtain data through satellite imagery processed by NASA for the years of 2007 to 2017. This helped us identify deforestation, degradation, and regeneration that was occurring in Brazil during a 1-year lag and a 5-year lag. The images depicted here show what land cover change looked like during a 5-year lag because those changes were more evident. But the same was done with the 1-year lag as well. Our environmental covariates were species richness of mammals, which was estimated in 2015, but we had as well mean precipitation and mean temperature rates, which were taken during each year of the study. But we adjusted for all of these factors in our adjusted odds ratio.
Here's our results table for our odds ratio values. We chose to look at odds ratios because it's a statistic that shows the association between two or more variables. After running full regression models in R, we were able to obtain our results. We found that hantavirus and leptospirosis were the only diseases to show significant results. For example, as regeneration increases over a 1-year lag, the odds ratio of hantavirus shows that there's a 104% increase in hantavirus cases, and over a 5-year lag, the odds ratios show that there's a 33% increase in hantavirus cases. Likewise, regeneration increases over a 1-year lag shows the odds ratio of leptospirosis that there's an 8% increase in leptospirosis cases, and over a 5-year lag, the odds ratio shows that there's a 27% increase in leptospirosis cases. Our results suggest that the dilution and amplification hypothesis are context-dependent in dengue fever and leishmaniasis. Additionally, our results also suggest that the odds ratios for hantavirus and leptospirosis closely align with the amplification hypothesis. Limitations for our project included underreporting, coarse spatial, and temporal scales for outcome data. Additionally, it's important to note that biodiversity data is available in a cross-sectional, so time-dependent effects are not captured. On behalf of Meredith College, our group would like to thank Dr. Perez-Hadrick for assisting us throughout this project and mentoring us.